Morning, everyone.
Good morning, ma'am.
Morning, ma'am.
Morning,
Good morning,
ma'am.
ma'am.
Good morning, ma'am.
Welcome back to a new week. Hope everyone's fine.
Yes, ma'am.
All right. Yeah. So yesterday, did you have the presentation? Afternoon.
Yes, ma'am.
Yes,
Presentation.
ma'am.
Okay. All right. Okay. So I think everyone has joined except for I think Bala. He's not there, right? All others are there.
Yes, Bala, Kumar, and Manivandan. Three of them is not there.
Okay, then we will just start the class then. Okay, so last class, we saw what kind of microorganisms are commonly found in all these industries and what kind of contaminations could happen, how the contaminations could happen. And next, we were supposed to see how to control these microorganisms, the growth of microorganisms. And as you all know that to have a completely or 100% sterile environment is impossible. So there's something called as microbial monitoring, or you can also call it as environmental monitoring. So in a lab setup, a lab, it's an environment. So in that particular environment, what kind of microorganisms can you find and how to control these microorganisms? So basically, these monitoring, okay? So all this is done by checking the quality of the environmental condition. Say it has to be maintained at a consistent factor. So you have to maintain the temperature, you have to maintain the humidity of room, so on and so forth. And monitoring completely every time you go into a lab and you keep checking. This is just to detect any kind of contamination. So what do? You go into a lab, you swab the surface, you streak it and check for the growth. That's what you do in environmental monitoring. So every few hours, you need to keep checking for any sort of contamination. It can be any type of microorganisms. So any change in the type of microorganisms or in the growth of the microorganisms, the number of colonies that you can find, when you take a swab, and when you swab it, number of colonies also. If there is a large number of colonies, then obviously it indicates more contamination. So all these, any changes in the consistent environment should be taken care of. Okay. So how many times do you need to do the sampling? Sampling is nothing but you are going and taking swabs from everywhere. So you will be assigned some area. Okay. So you are under environmental monitoring now. So you need to, as a microbiologist, you need to keep checking the contamination. So now we have this ISO standards actually. So ISO has been given some, so a clean room. Okay, they've been given different numbers. I hope you know all those numbers. 145755 something. ISO 145755. Do you remember that, or you've not heard of all those clean room numbers? Okay. So ISO has, given some standard numbers. And if those numbers are there, if it is an ISO so and so room number, that means it has to be maintained under certain conditions. That room has to be, maintained under proper sterile conditions. So if it is an ISO class 5, and I think class 5, class 0, class 100, class, I think 10,000 and all I explained previous class. You remember what is ISO class 100? Come on, people. ISO class 100, class 8, class 100. Only 100 particles should be present per square cubic meter. If it is more than 100, that means it is contaminated. Remember, I told you last class. Okay. So if it is a class 5, so in a hood, in a biosafety cabinet, it is called ISO class 5. That means not more than 5 particles should be present in that biosafety cabinet. So now every time you open and close the biosafety cabinet, or when a person is entering and working in that biosafety cabinet, obviously he's going to stir up the air. Remember, particles are freely present in the air. There are some particles. In a sterile room, there are some particles. Now, in those cases, you also have the HEPA filter in your biosafety cabinet. That is going to prevent the entry of the all these particles. It can be, when I say particle, it can be organic or inorganic particle. It can be anything. Okay. So now, once these, these particles try to enter. So if you are 2 or 3 people, actually you're not supposed to work in groups in a biosafety cabinet by chance. If 2 of y'all are there, one person is doing streaking, the other person is helping you out and arranging all the glasswares in the biosafety cabinet. Obviously, he is forcefully, making all the particulates enter into the biosafety cabinet. So that is how particles can enter the biosafety cabinet. Now, when I say class 5, that means not more than 5 particles per square meter is allowed there. And every time, so each shift, so maybe you are having a 3-hour shift or a 4-hour or a 6-hour shift, any number of shifts. So every shift, you need to take the swab. And you also have these air samplers. Now, actually, honestly speaking, you have many kinds of air sampling methods. You have some devices for that also. Okay, I think you must have seen in your labs also. You have some air sampling devices. So every, it's just that whatever particles are present, so they are collected on some kind of a sticky sheet, and that's actually made up of agar, onto agar strips. Okay, you collect the particles on agar strips and you test it. Test it for what kind of particles are present. It could be a microorganism or it could be any inorganic particle also. So how frequent do you have to keep checking for the particulates? So if it is an open space, you can keep checking it very often, maybe 5 times a day. Okay. If it is an active monitoring system, when you are doing any kind of surface monitoring, every evening or every day morning, you can come and check. And if there is any aseptic processing area, so in your processing area is where you are going to prepare medias and you are going to prepare all your glasswares. It's just your preparation room there, processing area. So here, each shift, you have to check for the contamination. And other areas, you can do it twice a week, and less critical area, once a week is fine. So that's an ISO class 8 room. So once in a week, you can go ahead and test it. Okay. So here you have another method. So one thing is active air sampling. Another method is the settle plate uh, exposure. So you are going to expose some plate, okay, containing some media, expose it for so many hours, maybe 4 hours or maybe 1 hour. You can even expose it to 5 minutes. I think even in your labs, you would have done when you were studying. So if you are a BSc, MSc student, definitely you would have done it. So just open media plate, expose it for 5 to 10 minutes. See, in an open space, you can expose it even for 1 minute also. You will have so many microorganisms in your bedroom or in your any room. But when you're talking about a sterile area, since the particles take time to travel, okay, whether they're organic or inorganic, they take some time to travel. So the exposure time is going to be longer. Okay, so you can expose it for 1 hour or 4 hours, maximum 4 hours you can expose it. Next is contact plate or swab. So everywhere in your biosafety cabinet or wherever you are working, your counter, your desk counters, your working counters, tables, take the swab, check. Okay. And your gloves, your garments, even your gloves, wherever you are touching, all those places also might be contaminated. You never know. You might think that I'm just wearing gloves and probably they're all sterile area. I can touch it. But even though you're wearing gloves, the area that you touch might be contaminated. Okay, so you need to follow these safety protocols to minimize the contamination. So here we have the air sampling, ISO 5, ISO 6, ISO 7, and ISO 8. So less than 10%, you have to report it as highly contamination. More than 10%, you have to report it as highly contaminating area. So less than 10% is acceptable. For ISO 7, less than 5% is acceptable. ISO 6, less than 3%. And ISO 5, less than 0.1%. So less than 0.1% means that's a perfectly sterile environment. Okay. Now, so all these are nothing but some agents which you use for cleaning the surface. This topic will be done by another faculty. So I'm just going to go through all the general things. So alcohol, the basic working principle will be done by another faculty. How it kills the organisms and all. So alcohol, generally everywhere you have sanitizers, which is made up of alcohol. Okay. Sanitizers, they work differently. So it's a general disinfectant or an antiseptic agent. Then if it is, if you're using chlorine or sodium hypochlorite, they're all spore killing agents. So any spores. So chlorine, definitely you use them in swimming pools also, outside. Okay. Now, in a lab, you can do chlorination. It's a halogen, which is a very, very, unstable halogen. It reacts, it releases a lot of free radicals, which goes and, I hope you understand free radicals, right? So it releases a lot of free radicals. All these free radicals are unstable. They will have to combine with other gases to form stable compounds. That's how oxidation takes place. Then phenolics. Okay. So phenols, generally you use them as disinfecting agents. Ozone, again, it's an oxidizing agent. Okay. Hydrogen peroxide, also an oxidizing agent. And other, you can have substituted diguanides. There are also antiseptic agents, then parasitic acid, ethylene oxide, very commonly used again, and quaternary ammonium compounds. Again, these can be also used, and lactone, beta propiolactone. So all these are some spore killing agents, which is generally used. So, how does all this work? So what does environmental monitoring achieve? What do you achieve with this environmental monitoring? So, and where all can this be done? So you can do environmental monitoring in media fills, in rooms where large amounts of media is prepared. Disinfection regime, always carry out the proper, maintaining of disinfection has to be carried out if you want the lab to be under sterile conditions. Then any kind of environmental deviation. So if there is a contamination, obviously, this is possible because of the irresponsible people or irresponsible, if by chance. Then HVAC, utilities, then QA/QC, personal cleanliness, then any kind of facility room, okay, and aseptic processing line, then personal flow, material flow, or any kind of layout. All these areas we follow environmental monitoring. So, the FDA now. So this FDA also pressurizes the industry that in a class 100 area, no microorganism has to be detected. You can detect inorganic particles. They are not going to cause any contamination. But I should not detect any kind of microorganisms, nor any kind of spores should be present. Then any product, the product contact surface, supposing you're manufacturing medicines, tablets. So how do you think the tablets come out of the unit there? They'll have to be on some surface, right? Before they are packed onto before packaging. You've studied packaging, right? Before, into aluminum or plastic sheets. Before they are packed into all these also, those contact surfaces also has to be sterile. So the plastic, whatever the packaging material, also has to be in a sterile condition. Then media fill. So any media that you're preparing and filling those medias, they should be free of contamination. And all these have to be checked. So if there is any abnormal deviation or any kind of deviation in depth, is gone. So that entire batch of tablets can be discarded if there is contamination. So these are the recommended limits for microbial contamination. So in air, it is CFU, that is colony forming units. CFU stands for colony forming units. So you should have less than 1 CFU per meter cube. That means in such a small area, less than 1 colony or no colony should be there. Okay, and settle plate. So you're taking a plate of certain diameters. So those diameters, so in those plates, the CFU should be less than 0.9 mm. So this is for a time period of 4 hours. So within those 4 hours, if there is any bacterial growth, it has to be less than one colony. And contact plate, and even when you're wearing gloves. So after you wear the gloves, after you work or during your working process, the 5-finger gloves, just press them on a plate, on a media plate, and check for the colonies. So even after wearing gloves, there is a probability of contamination. Okay, and so these are all the ISO standards. So ISO class 1, class 2, class 3 up to class 9. What are the limits? What are the concentration limits? You can just go through them in the slide. And yeah, so how can this environmental monitoring be achieved? So we have these aseptic biomanufacturing environments, so which requires a strict, control. Just a minute, I'll show you another thing also. So any kind of contamination or all these, so if you are practicing GMP, so there's supposed to be a lot of, the product consistency has to be maintained. The products has to be safely maintained, purely maintained. So how do you achieve all this? So one thing is by using filters. So you can, whatever equipment you're using, okay, it can be a laminar airflow, you can use a HVAC system, or any ongoing HEPA filter program. So if you're in a room, obviously the room is completely, when it is a sterile room, you have HEPA filters also, right? So all these HEPA filters, you need to check if there is any kind of leakage in the filtration process. So you need to perform a airflow test. So whatever air is coming out of the HEPA filter, you need to check that. Okay. So all these verification has to be done, and clean room cleaning, any kind of disinfecting processes can be used. And how do you qualify clean room qualification? So before you use the clean room, any area you need to check. Okay, you need to check for the cleanliness. And this qualification is also called as PQ. So you need to check the consistency. You need to extensively go check for the cleanliness there. And also, sometimes if there is a power outage, if there's a power outage, the HVAC system is not going to function. So in that case, how would I maintain the cleanliness of the room? Even if the power goes off for like 5 minutes, the room is still going to get contaminated. So how do I maintain the cleanliness of the room? So next is the airflow pattern. So, whenever, the air flows out of the HEPA filter, obviously, when the air is flowing out of the HEPA filter, it flows only in one direction. So now, if there is any kind of leakage in the HEPA filter, so the direction of airflow is going to change, right? So that is how we can detect. So you can add some kind of smoke, okay, to check for any kind of leakage. And if the HEPA filter is working correctly, and if the airflow is just flowing in one direction and not in multi directions. And also, so this airflow pattern has to be checked at least once a year. So once a year, you have to check for the leakage and all. Then you have SOPs, that is standard operating procedures for the performance on environmental testing, documentation of the result. So, and you know that documentation is very, very important, right? Even if you miss one single point, that is going to create a lot of conflict. So what you have to do is, even by chance, so what colonies that are growing. Okay. So when you do the swabbing or when you do the exposed plate method, immediately you have to note down or you have to document the number of growth of the organism. If you miss even a single colony, then in your documentation is going to go wrong, and this can lead to some very bad consequences. Someone is having their mic turned on. Then everyone has to be trained for this environmental monitoring and testing procedures. So the training can be conducted by performing any of these tests. So I hope when you are doing your BSc or your MSc or even MPharm, whatever is your combination, basic techniques are microbiological techniques. So how to take a swab and how to expose the plate and how to check for the different colonies. All these are some basic training and training you have to undergo. And what are the different components of a very effective environmental monitoring program? So when you start any kind of manufacturing facility, whether it is drugs or whether it is anything, okay, even if it is food also. So you have to keep monitoring all your environmental conditions continuously. And airborne particles, these airborne particles, whether they are viable particles or non-viable particles, they can cause any contamination level. It can be a high level or it can be a low level contamination also, because it depends on the pathogenicity of the organisms. Okay. And any kind of contamination in clean utilities, in a clean area. And all these factors you have to maintain: the temperature, the humidity of the room, the direction of the airflow, then pressure. All this matters a lot for your environmental monitoring program. Next, I think this I already told you. Where all can you collect the samples from? So collecting of samples, you can do it from wherever, whenever. Okay. So any thing that you keep coming in contact with very often. So that's the first thing that you have to check. So if you are doing some aseptic culturing or if you're doing some aseptic manufacturing, so any instrument that you keep coming in contact with, those instrument surface, check for contamination. So do a swab there. Then near the product, so any area near the product, that is the most important area. So check the swab there. Then any area of high personal and equipment flow. So near the HEPA filter, near your autoclaves, all those areas you can check. And what is the particulate level or the microbial level? So that is on you, on the equipment, all this can be checked. Okay. Then these are the most critical sites. So in an LAF, in a laminar airflow unit, that is in your whatever biosafety unit also. If the sample within 1 feet, so usually your LAF is about 1 feet by 1 feet. It's a small area where you can perform all your media preparation. You can do your medias, you can prepare your plates and things like that. So here, any increased activity in this particular unit has to be controlled. So keep sterilizing this area very often and surface contact plate. So any area that you keep coming on the surface. So for example, the tabletop that you work on, the product containers, okay, the processing unit there, all those are the surface contacts that you need to check if it is contaminated or not. Because these are the areas that are frequently getting exposed to. So these frequently exposed areas are the ones that you have to keep checking very often. Okay, and then the area where you wear your gowns. Okay, so basically your forearm, your chest area, your gloved fingertip. So all these areas has to be checked frequently for contamination. Okay, so supposing the contamination can happen two ways. One, without knowing, you are carrying the contamination. You yourself might not know. So if you are having some kind of an infection or if you are having some kind of a flu, you have a lot of microorganisms in your body. Obviously, you're going to contribute to the contamination. So do not go there. You do not go to work if you're having a flu or a cold or a cough, or if you're having fever. Okay, do not enter into the sterile areas there. Or, so this is you contributing to contamination. Another reason, another way of contamination can be you without knowing yourself, you are carriers of contamination. So you're working somewhere, okay, you come in contact with some microorganisms, you will not realize that. You go to the next area, okay, you're carrying the contaminants, that is microorganisms, from one area, from one sterile area to another sterile area. Okay, so the gloves that you're wearing could have been contaminated by the microorganisms and you without knowing going and touching the surface area, other surface area. So this can lead to transfer of all these organisms. Okay, so all these are some examples of contamination. So here in a class 10 area, so what is the amount of microorganisms? So microbial air once per process. So if it is an active area, so that is active area is nothing but where you frequently carry out your work. So passive area, you don't visit there frequently, but maybe say once in 3 hours you go into that area. Okay, so that is passive area. You can check, you can just place a plate there, microbial plate, and you can check for the growth later on. Then surface contact plate, minimum at least one site per process. So these are the areas where you frequently keep, going in and touching all these surface areas. So check for the fingerprints. I think this I've told you all many times. Okay, and particulate air. So minimum at least once per hour, you need to keep checking the particulate air. Okay. So now I think, so this is the graph that is going to show you what is the three different categories of environmental test results. So if you have very high amount of contamination, that is at say 120, so this is the number of colonies there. So if you have these many number of colonies in any kind of condition, then you need to take action. That is, you need to completely sterilize yourself. You need to sterilize the gowns that you're wearing, okay, and the complete area. Then the yellow one, that is approximately 90. Okay, if you have 90 colonies, it's an alert. You need to be careful. You can start with the or you can start preventing the contamination. And acceptable range is maybe less than 0 or 1 colonies. Okay, 0 or 1 colony, it is acceptable. More than 90 colonies, it's an alarm. So you need to be careful, prevent the contamination. Okay, so these are for the particulates. So particulates are all the inorganic uh, particles. It can be hair, it can be dust, it can be all these, any salt also, you can have salt particles in the air. So less than, in a class 100 area, less than 3,520 particles. Class 10,000, that means per square meter, I'm going to have approximately only 10,000 particles, not more than that. Okay, so these are the different, so class 100 again, 100,000 particles per square meter. That's why it is called as class 100, 10,000 and 100,000. So in a class 100, it is permissible. It is permissible for at least you can have 3,500 particles, approximately. Okay, and then the number multiplies exponentially for the different classes. And any CFU or any colony forming unit, any microbe, if it is active, in a class 100, less than 1 colony. In class 10,000, less than 10. And in class 100,000, less than 100. And in a passive microbial air, you are not supposed to find any kind of, you can find, okay, colonies, but the number is very, very less. So surface contact plate. So in a surface contact plate, you can find less than 1 or 5 or 25. And next is floor contact plate. The most important, because you're going to walk on the floor. Okay, you're going to completely cover your shoes and slippers as well in a completely sterile area. So the floor also has to be monitored. And another important factor that I didn't tell you till now is compressed gas microbiome forming units. So compressed gas. Now, you are aware that any kind of cell culture requires carbon dioxide. Okay, any anaerobic organisms, they are also going to require air. So you're going to, and in a fermenter, now when you are preparing media, okay, for any kind of fermentation process, you need to pump in sterile air, sterile oxygen, or it can be oxygen or carbon dioxide. Now, how are you getting the carbon dioxide and oxygen? One thing is you get in cylinders, right? But do you know in that cylinder whether there is any kind of microorganism? There can be spores in them. Okay, now I need to check whether those compressed gases are also free of microorganisms. Okay, now I need to check them also. So even those gases, you need to expose it. You can do an exposed plate method only with those gases also. So you can check for any kind of spores or any microorganisms. Okay, so so these are certain microorganisms that I have given. So these organisms, if they're all like they come under high radar, okay, by FDA. These are the microorganisms that are commonly present. They are all pathogenic microorganisms. And when you are manufacturing any pharmaceutical product, these are the primary contaminants. So you have to know the colony characters of Burkholderia sepia, Escherichia coli, Pseudomonas aeruginosa, Salmonella shigella, and Staphylococcus aureus. You have number of species, subspecies. You have number of variants of all these. So you need to know the colony characters of all these microorganisms. So whoever is into environmental monitoring, these are the organisms which you can find as a common pathogenic contaminants. Okay, now, and these organisms, they have their own features. For example, E. coli. If there is fecal contamination, you will find a lot of E. coli. Okay, poor production hygiene, you will get Staphylococcus aureus. Staphylococcus aureus, you will get it in the air also. Okay, so all these are, so these organisms can act as indicators also for contamination. So these are all objectionable organisms. Whether how many ever times you sterilize it, you'll definitely find those organisms. And there are certain elements that you have to keep checking with. So how many you can identify, or you have to keep reviewing the samples, you have to keep, for that particular location. So if you, on the countertop or on the floor, if you get more than 100 CFUs, then you will call the cleaning person, or you will recommend, you put a tick for cleaning that the room has to be cleaned immediately. Right? That's how you do it in the industry. So immediately check, clean the room, clean the area. After cleaning is done, again, you'll have to do the review. Review is nothing but take the swab wherever the area has been cleaned and check for the growth. Okay. And you have other parameters that you maintain in a sterile area. So your airflow, the pressure, all these has to be maintained, and the pressure or the airflow that you are doing, check whether it is free of spores or microorganisms. So these are all some, agents that you have to keep checking every now and then. So when you take precautions in these areas, you are going to eliminate contamination. So during your, your working in your, if you're working near the biosafety cabinets, restrict the movement of people. Don't walk around the biosafety unit. Come, sit and work, and no talking. Okay? Only actions. You have to learn sign language. Okay. So, yeah, only actions, less communication using your mouth, and HEPA filters have to be tested for leakage. Then keep disinfecting the area with sporicidal agents. You are going to reduce this bioburden. Okay. Bioburden is where you are going to cause a lot of contamination. So personal training, personal hygiene has to be maintained. Cut your nails. Okay, do not grow nails. Do not apply nail polish. Okay, make sure your hair doesn't stick out. You have to wear the cap. Okay, so no exposed hair. You have to keep your all your exposed skin surface has to be covered, and keep cleaning the hoods. If there are different designs, I think LAF has been done, right? Earlier. Yeah, so you have different designs of biosafety cabinets. So all those, maintain it, keep checking for contamination, check the airlocks, okay? And any items, unnecessary items which you place in the biosafety cabinets has to be removed. So all these are certain rigorous methods that you need to follow. When I say rigorous or critical, it means that if at all, if you remain careless in any of these areas, the entire production of whatever you are manufacturing is going to go for a toss. Okay, so it can be very critical to you also. So you have to focus and you have to maintain all the records properly. So how many colonies that that has formed, and you need to show the plates also to your supervisor. So you cannot randomly say that I have only 2 colonies where you have 4 colonies, because all those plates you need to show to your supervisor. Okay, and these are all the different sampling methods. So sill to agar air samplers, STS. You have a unit which is attached to a vacuum. So the air is sucked in and it is transferred to a petri dish containing media, and any airborne particles, all these will be sucked onto this and plated onto the plate then. So this is still to agar air sampler. Then you have sieve impactor. So it's a very small dish. It's a very small unit that can, accommodate a petri plate. And again, you have a vacuum pump that is going to draw air from outside. And again here, similarly, it is going to stick onto the all the air particles, or the air is transferred into the plate there. Okay. And both, they're very similar. The principle is same. Sill to agar air sampler and sieve impactors, they're the same thing. Okay, but only the design is different. Then you have other type of sampler like centrifugal sampler. So you have a propeller. So like a, wind turbine. You must have seen propellers in your bioreactors and all that. In manufacturing units when you have gone. These are, or in a boat. You've seen on the boat, the propellers which is used to navigate the boat. It's used to push the boat forward. So these propellers are present and they pull in the air, and you have nutrient strips placed at the base of the unit there. So all those forcefully pulled air, whatever particulates are there, they will stick onto the agar strip there. So these are some examples of air filters that you can use. Okay, so settling plate, again here, you need to expose it for 4 to 5 hours. Okay, it's just, you can prepare these plates, media plates, keep it wherever, possible for 4 to 5 hours. And also, you need to maintain the particulates also, right? How do you maintain, or how do you remove these inorganic compounds? So you have certain static testing facility. So static test, dust is attracted to static electricity, right? You have done these experiments even when you were kids also. You take a scale, a plastic scale, you rub it on hair or you rub it on some wool, you create static electricity. So dust and paper bits and everything sticks onto this statically charged surface, right? So you have some static controllers. So all these static controllers, you can just switch it on. It is going to absorb all the particles. Okay, and you can remove that static machine, and you can release all the dust particles. Because if you leave it in the room, then once the static electricity dies out, all the dust particles will be again released in the same way. Okay, so you need to switch it on for some time, remove all the static particles or the dust particles, and clean it off. So clean this unit then. So I think air monitoring, I told you already. So any airborne contamination, So air monitoring, you have to do it for both viable contamination and non-viable contamination in the clean room. Then microbial air monitoring, so you have different samplers. All these you have to keep checking. And active microbial air monitoring. So we have different samplers. So these are some examples. So all this which I told you earlier, SIL2 agar, centrifugal air sampler. So these are all the pictures that I could find. So this is non-viable particle counter. So this is your static electric counter. Okay, you can use it to count the particles. And these are some surface equipment monitoring system. Okay, so you have your agar plates. So these are all the colonies that you see in white there in your in the Petri plate. Okay, so how many colonies are there? Can you count and tell me how many colonies are there?
5.
5.
There's actually 6 colonies. You missed out the one small one towards the left. Can you see this? If you can see the pointer. So this 1, 2, 3, 4, 5, and 6 colonies. So 6 is a permissible amount or no? Is it under a permissible limit? The number of colonies, they seem to be very big actually. Now, one more thing, the size of the colony also matters. Okay, because that one colony will not have only one organism. They will have millions of organisms in that one colony there. Okay, so the size of the colony also matters for the contamination. So this is just your microbial monitoring of gowns. So wherever you keep getting exposed to these contamination, that is all your 5 fingertips, the gloved fingertips, the chest area, okay, because this is where you are. So whenever you're working in the LAF, your head is covered, right? In your biosafety cabinet, head is covered. So this chest area is where you are exposed to. So this area is also checked for contamination. Then utility monitoring. So this is another important feature where all the utilities, that is the water that you're using to prepare the media, the air, carbon dioxide, all the gases that is supplied, okay, the compressed gas, steam that is also supplied, everything has to be maintained in a sterile state. Okay, so these are all the areas that you have to also perform the environmental monitoring. Okay, and why do we need to do this environmental monitoring? What are you gaining from that? So from all this, we are gaining knowledge. What kind of microorganisms can cause contamination, how to reduce the contamination, and how to prevent the contamination. So this is called as knowledge management. So we need to practice this knowledge management, okay, to prevent any kind of contamination. So we have some risk management also. You can, so all this is a part of your, I think this someone else is going to be taking this risk management and quality risk management and all that. Okay, so I have just given a few definition. So any product, any process that is manufactured, which is commercial or which is of commercial importance, you need to have the right approach and have the right knowledge for the processing and understanding of that particular product. And knowledge management, that is defined as acquiring data, analyzing, sorting those data, and leaving out all the unrelevant data. Okay, the manufacturing processes, the product of the product and everything. So all these are nothing but your knowledge management. Okay, and knowledge management is very, very important in environmental monitoring. Okay, because you're collecting large amount of data. So your data is nothing but in environmental monitoring, how many colonies. Basically, it's all the number of colonies that you collect. What type of colonies they are, the color of the colony. Okay. Then you have to test for the microorganisms. So who is working with in that particular area? Okay, which person, which employee is working? Who, so all these will be collected and so that is all your data. And quality control practices. So this is again very, very important because this is important for eliminating any kind of contamination. Okay, so quality controlling is also another important factor. So do you all have any questions related to environmental monitoring and microbial contamination? Yes, Prashanti, any question? I hope I didn't bore you all. Early morning. Prashanti is laughing. What, Prashanti? Feeling sleepy. Yeah? Okay, so if there are no more questions, then no questions? Okay, then we will take a break now. So by 10:15, we'll meet. We have another faculty, Mrs.
Hello. Welcome to our next session. Is everyone there? Everyone joined? Okay. Next section is about fundamental and non-viable and viable particle count and importance of monitoring. And the speaker is Ms. Soumya Shanbhah. I would like to introduce our speaker. Everyone are joined, right? Everyone are there, no? Can you hear me?
Yes, sir.
Yes.
Yes, sir.
Okay. Ms. Soumya Shanbhah is currently working as a head and assistant professor in MS Ramaiah College of Arts and Science Commerce, Department of Microbiology, Bengaluru. She has 16 years of teaching experience, including the role of assistant professor, and her expertise includes biochemistry, molecular biology, and industrial microbiology. And she has several distinct publications with different esteemed journals. And she also been recognized as a best teacher award from Om Shri Narayana Charitable Foundation. And also she has served as different resource person for several workshops as well as the webinars. And we welcome you, ma'am.
Thank you, sir.
Yes. You can start now, actually, everyone. Are there.
Okay, okay. Good morning, everyone. Welcome to the next session for today. I'll just share my screen, then we can start the session. Is the screen visible to everybody?
Yes, ma'am.
Yes, ma'am.
Yes. So you are doing this course from so many days and you are learning about many aspects of sterile manufacturing practices in different industrial perspective. So today we are going to start with one small point, small topic in this, which is the fundamentals of non-viable and viable particle counting and its importance or its importance of its monitoring. So we'll start with the introduction. What is mean by viable and non-viable particles? So, and what is mean by environmental monitoring? Now, when you go to any industry, it is very, very important to keep monitoring the areas in the industry or different areas in the industry. So this environmental monitoring, it is going to give you it is going to describe the process or the activities which has to be conducted, which should be conducted to monitor the quality of the environment in the industry. Are you getting, everybody? Are you understanding the what is mean by this environmental monitoring? It is the process which is going to determine the activities or need to characterize or monitor the quality of the environment in the industry. Or in another words, we can say this environmental monitoring is a surveillance system which will determine, which will understand what are the microbiological microorganisms or microbiological aspects and non-viable particles present in the environment of the industry. Now, there are different kinds of environments. Now, this process is going to provide the monitoring criteria. It will help us to find out how we are going to test this viable and non-viable particle counting. And it is going to give us the feedback on microbiological quality in different levels of the aseptic environment. Now, what are the different areas where this has to be monitored? So it can be clean room areas where the processes are happening. It can be sampling areas where in the industry different sampling techniques are carried out, or in any other area in the industry, this particular counting has to be done. Viable counting as well as non-viable counting. Now, when it comes to non-viable counting, what you will understand with non-viable counting, you will get to know how many particulate matter, foreign particles are present in that environment. When it comes to viable part counting, what you'll know about the sterility of that particular environment. What is the count of microorganisms? How many microorganisms are present in that area? So with these two monitoring, non-viable particle monitoring and viable particle monitoring, we will understand the foreign and particulate matter present in the environment, and second, the sterility. So these are the two important aspects which we will know when we count this viable and non-viable particles in an environment. Now, what is the need? Why we need to calculate? Why we need to enumerate this counting, or why we need to count these particles? This particular process is going to provide us the information about the quality of the aseptic processing. So in an industry, when the processes are happening, say for example, in an industry where R&D process is happening, research and development is happening, during that process, the aseptic environment should be maintained. So this viable, non-viable counting is going to provide us information on the quality of the aseptic processing, which is happening in the manufacturing environment. First thing. Second, why we need to do this one? It is going to prevent the release of potentially contaminated batch. Say, for example, any food product or any pharmaceutical product is going to be released in the market. And if we have not done the counting, this monitoring is not happening. So what can happen? There are chances that the product which is released is contaminated, potentially contaminated batch. So if this counting monitoring is not done, there will be release of contaminated batch. So with this monitoring, we can prevent the release of potentially contaminated batch, which is going to affect the end user. Then it is also going to help us to prevent the future contamination. Because if one batch is contaminated, there are chances that the next consecutive batches also may get contaminated. So this environmental monitoring for viable and non-viable counting will provide us information about the quality of aseptic processing. It is going to prevent the release of potentially contaminated batches, and it is going to prevent the future contamination. This is the reason why we need to monitor this viable and non-viable particle count in the industry. Are you getting? Yes?
Yes, ma'am.
Now, what are the different areas where in all we have to do this counting? In an industry, it is a huge area. Which areas has to be controlled or which areas has to be taken into consideration when we are doing this counting of viable and non-viable particles? So the area includes clean rooms. So the clean rooms are the major area where this viable, non-viable particle counting has to be done. Then the formulation tank rooms, where the different formulations are happening, those tank rooms has to be monitored. Laminar air flows, or we can say the biological safety hoods, where the research is happening and many experiments are happening during this industrial processes. So these areas has to be monitored. Then the isolators. So when the product is taken from one place to another place, one clean room to another clean room, so those isolator areas, whatever isolated areas are there, those also has to be covered or monitored. Then intravenous compounding areas, when packaging system is happening, when the product is getting packaged into sterile containers. So during packaging, during intravenous compounding, all these areas have to be monitored for viable and non-viable particles. All right? Is it clear till here? Yes? Yes or no?
ma'am.
Yes. Now, first thing we should know is what is mean by this aseptic environment. Till now, I have told you that aseptic environment 2-3 times I have told this word. Now, what is mean by aseptic environment? So aseptic environment means it is going to be the area which is free from contamination. Now, contamination from what? Contamination from harmful bacteria, viruses, or any other microorganisms. All right. Now, what is the need for maintaining this aseptic environment? So to keep the place clean wherever this industrial processes are happening. So it can be clean room, or it can be the manufacturing area, or any kind of area where this entire industrial process is happening. It has to be aseptic environment so that there will not be any contamination, microbial contamination into the products. So why this microbial contamination is important? Because this microbial contamination, if it is happening in the product, it is not only going to hamper the product, quality of the product, but it is also going to affect the end user. So end user is affected. So it is very, very important to monitor this contamination. So we have to prevent the contaminants from entering, from growing in any of these products. So what are the different particles which we are going to count? We are going to count the non-viable particles and viable particles, which is essential for quality, to check the quality of the product, safety for the end users, as well as for the person who is in the process of manufacturing this particular products. So quality is for the quality, maintaining the quality, for the safety, and for the compliance in the industry in different applications. So for all these reasons, it is very, very essential to monitor viable and non-viable particles. Now, what is mean by viable and non-viable particles? So when it comes to non-viable particles, it is the particulate matter, because in an air, there will be lot of dust particles, skin cells, because lot of persons are working in the industrial areas. So there are skin cells which are liberated and they will be in the form of aerosols. So clothing particles, pollens, etc., etc. So any non-living particulate matter which is present in the aerosol as an aerosol in the environment, it is considered as non-viable particles. So if you are going to count those non-viable particles, the process is called as non-viable particle counting. Now, this non-viable particles, what is the basic difference between viable and non-viable particle? These are non-living particles, and hence these particles will not grow or spread. Right? But still, these particles are going to possess, they are going to have a serious threat to an aseptic environment, because indirectly they are going to affect the aseptic environment. And hence, these non-viable particles has to be counted. Now, how we are going to count these non-viable particles? By a simple technique of laser counters. So laser counters will be installed in the different areas of the industry, and the particles will be counted. Now, what is meant by viable particles? Viable particles include living microorganisms. Now, living microorganisms means which microorganism we are talking about. It can be yeast, it can be bacteria, it can be mold, it can be fungi, and many more. So whatever living microorganisms are there, those are included in viable particles. Now, what is the difference between non-viable and viable particles? Viable particles are having potential to grow. So they are more dangerous because they can grow, they can increase in number, and they can contaminate the other areas also. So non-viable particles are the particles which cannot grow, which will not grow or spread. And viable particles are the living microorganism which can grow, which can spread to the other areas. So now in viable monitoring, what we are going to do? We are not only detecting the organisms, these viable particles, but also we are going to enumerate this microorganism. Enumerate means what? Can anybody tell me? What is mean by enumeration? Yes? Can anybody tell me what is mean by enumeration? Detecting means we are going to just detect whether the microorganisms are present or not. But when I say enumeration,
Counting.
yes, exactly. Very good. So we are not only going to detect the microorganism, but we are also going to enumerate or count how many microorganisms are present in that area. So this is about the aseptic environment. Now, what are the different sources from where this particles will come into the environment? So it can come from the air, because in the industry, whatever air is circulated, the particles, viable as well as non-viable particles, can come from the air. It can come from the person who is handling the equipment, who is present in that area. So it can be coming from the personnel. It can come from the equipments which are used. So this contamination can happen from the equipments. It can happen from the cleaning agent, whatever agent, different chemicals are used during the processes. So it can come from the chemical agents or cleaning agents. Then the containers which are used by the industry or during the processes. So the contamination can come from containers. Second important thing is water which is used during the processes, industrial processes. So contamination can happen from the water and from the compressed gas which is used in the industrial processes. So these are the different areas or different sources from which there are chances of contamination. It can be air, it can be the person who is handling, or it is the person who is present there, then the equipments, chemical agents which are used, containers which are used, water, and the compressed gas. Now, when we are enumerating, when we are counting, when we are monitoring this viable and non-viable particles, what are the specifications, or who is given the specification for this acceptable particle counts? So there are different regulatory bodies which has given the acceptance level of this particle count. Which are those regulatory bodies? So depending upon the different industries, it can be Food and Drug Administration, that is FDA. It can be International Standard Organization, that is ISO. There are different ISO standards they have given. Then Parental Drug Associates, that is PDA, European Union, and United States of Pharmacopoeia. So these are the regulatory bodies who has given the standards for viable and non-viable particle counting. All right, till here, is it clear for everybody?
Yes, ma'am.
Yes?
Yes, ma'am.
Yes, ma'am.
Okay. Now, what is the criteria for this monitoring? How we are going to monitor this viable and non-viable particles? So, as I told you, there are different regulatory bodies which has given the standards. So, what are those standards? What are those criteria for this counting? So, some areas in the industry has to be monitored for this viable and non-viable counting daily. So, it is called as daily monitoring areas. Which are those daily monitoring areas? The laminar airflow, UV pass box, and the cleaning rooms. Now, what is meant by UV pass box? Can anybody tell me?
Transfer of
that-
material from one region to another.
Exactly. So when a product is taken from one clean area to another clean area, or one region to another region, the passage with which this is going to be transported, that particular area is called as UV pass box. Now, this area is very important because laminar airflow hoods or clean rooms will be maintained with all the air pressure, air velocity or not. When the product is getting transferred from one clean room to another clean room, the passage, which is called as UV pass box, this passage also should be monitored on a regular basis. So daily monitoring is required. Otherwise, there are chances of contamination during the transportation. And when there is changes in the air pressure, air velocity, so there will be problems. Many problems will be related to the product. So daily monitoring. So LAF has to be daily monitored. UV pass box has to be monitored, and the clean rooms has to be monitored on the daily basis. Now, what is the monthly monitoring? Once in a month, what kind of areas will be monitored once in a month? So manufacturing area, where the process of manufacturing is happening, the slabs, the tables, whatever that entire area is there, those areas has to be monitored on a monthly basis. Equipments which are used for the manufacturing processes has to be monitored on a monthly basis. Then the drain points, the outlets where the water which is used or the water which is getting in, so those drain points has to be monitored on a monthly basis. So daily monitoring, monthly monitoring, next comes quarterly monitoring. So the compressed air which is used in the industry has to be monitored on a quarterly basis. Half yearly monitoring, the operators or personal hygiene. Now, personal hygiene means the person who is handling the equipments, who is handling the manufacturing process. So personal hygiene of the person has to be done on a half yearly basis. So we'll see what are the different personal hygiene monitoring later when we are seeing the viable and non-viable counting. Now, yearly monitoring. So for yearly monitoring, the system which is given is called as HVAC system, which is nothing but heating, ventilation, air conditioning system. So this heating, ventilation, and air conditioning system is going to affect the efficacy of the material, raw materials which are used. It is also going to affect the in-process goods and finished products. Hence, this HVAC system is very, very important during the processing, during the monitoring. So it is called as HVAC validations. Then there is something called occasional monitoring. Now, what is meant by occasional monitoring? If the laminar airflow filters are not used, or they are not they're not working. So during the maintenance, during the change of the filters, so all these monitoring, whatever is happening, those are called as occasional monitoring. So in an industry, different areas will be monitored for viable and non-viable counting on different basis. What are those different criteria? So it can be daily monitoring, monthly monitoring, quarterly, half yearly, yearly, and the occasional monitoring. So there are different areas in the industry which will be monitored on these bases for the viable and non-viable particles. Now, what specimens we are going to collect from which these areas, whatever areas we have seen, laminar airflow or the manufacturing area. So whatever areas we are seeing, which kind of specimens we are going to collect to monitor the viable and non-viable particles. So it can be air, it can be surface, that the floors, walls, or the equipment areas. It can be personnel, that is the person who is handling or working personnel. So the personal, the specimens will be collected from the person. Then the drains. So whatever drains are there in the manufacturing area, though from those places, specimens will be collected. So to count particles, that is viable and non-viable particles, we are going to collect the specimen, collect the samples from the air, surface, that is the walls, floors, and etc. Then the personnel, because the person who is handling the equipment, and the drains. These are the four areas, four specimens from where the samples will be collected and it will be monitored. Now, the first important thing, air quality. Sorry. Now, how we are going to calculate the air quality? So from air, how we are going to take the sample? Now, there are two ways to collect the sample from the air. One is called as passive air sampling, and another is called as active air sampling. Now, what is meant by passive air sampling? We'll be just exposing the plate in an area and allowing the air to settle, allowing that particular area air to settle on the plate. And we are going to use that plate, which is called as plate exposure method. We are going to use that one to count the viable organisms, that is microorganisms. Now, when it comes to active air sampling, now what is meant by active air sampling? You are going to force the air with some pressure on the plate. So you know how much air you are pressurized, forcing on the plate or the area, and you are going to count the microorganism in that particular area. Now, for this active or passive air sampling in industries, we are going to use the plates. Now, to count the viable microorganisms, what we are going to use? To count the viable microorganisms, we are going to use the media plates for growth of microorganisms. Do you have any idea about the media plates? What is mean by media plates? Yes? Can anybody
Yes,
tell
ma'am.
me? Can anybody tell me example name of any media which you
Nutrient.
know? Nutrient agar.
agar.
Yes, nutrient agar, nutrient broth, MRBA. So what is mean by media? It is the environment, it is the growth conditions which we are going to provide for the microbial multiplication. So microorganisms will grow by utilizing the nutrients which are present in that media, and they will form a colony. Now, bacteria. Can we see the bacteria in a naked eyes without microscope? Can we see
No,
the
ma'am.
bacteria? No. But on a plate, we can see colony of bacteria, right? So we are going to use some plates. So in industries, there are different plates which are used, media plates. So the media plates are called as Rodak plates. Now, the size of this plate for air sampling is 55 mm. Sometimes 90 mm plates will be used. So depending upon the industry standards, different size of the plates will be used. So now, most of the industries uses 55 mm Rodak plate. Now, what is meant by this Rodak plate? It is a plate where the organisms are going to replicate and they are going to grow and form a colony. So Rodak means replicate, organism, detection and counting. So when organisms are replicating or when organisms are multiplying, forming a colony which can be detected on a plate for the counting process. So these Rodak plates, depending upon the industry, there are different types of Rodak plates which are used in the industry. What are those different types of Rodak plates? One is S type of Rodak plate, E type of Rodak plates, and T type of Rodak plates. Now, what is mean by S type of Rodak plates? Now, when we do this viable counting means we are counting for bacteria, we are counting for fungi and other microorganisms. So in industry, there are certain microorganisms which are given more importance. What are those microorganisms? For example, here I have given one example, Staphylococcus aureus. Now, what is the Staphylococcus aureus? This is the organism. Can anybody tell me it is a gram-positive organism or gram-negative organism? Staphylococcus aureus. Have you heard about this bacteria?
Yes, ma'am.
Is it gram-positive or gram-negative?
Gram-negative.
Gram-negative. Anybody else?
Positive,
Anybody?
ma'am.
Gram-positive. Staphylococcus is a gram-positive coccus, cocci shape. Okay, gram-positive organism. Now, why we are giving importance to this Staphylococcus in the industry? Because of two main reasons. What are those reasons? One, the Staphylococcus aureus is one of the causative agent, or it is the organism which is present on the skin of human skin, which is also a causative agent for skin infections. So Staphylococcus aureus, what is the normal area where this Staphylococcus is found? It is normally present on the human skin and it is a causative agent for skin infections, wounds or any skin infections. Now, why we are going to count for this Staphylococcus? Because the person who is handling the equipment or who is involved in industrial processes might be the source for the contamination of Staphylococcus aureus in the product. So we need some specific media to check, to count the Staphylococcus aureus. Now, what is that media? So this media is called as BPA media. What is BPA media? Baird-Parker agar. So this is a special media which is going to detect Staphylococcus aureus. All right, we will not go into details of how it is used for Staphylococcus aureus and all. It is a specific media for Staphylococcus aureus where Staphylococcus will grow and form black color colonies on this Baird-Parker agar because of the ingredients which are there in the media. All right. So what is the reason why we need to monitor Staphylococcus? One, it is the causative agent of many skin infections. So it is present on the skin. So it can be the person who is handling can be the source of contamination. Second, the Staphylococcus is one of the organisms which produces toxin. All right. The Staphylococcus produces toxin, which will be harmful if it is produced in the product. If the Staphylococcus is getting contaminated in the product, and if the Staphylococcus is there in the product, it can produce toxin, which can cause food poisoning for the end user, if the end user is consuming that particular product. So it is very important to monitor Staphylococcus aureus. So for Staphylococcus aureus, we are going to use this Rodak plate, which is what type of Rodak plate? S type, that is Baird-Parker agar. Now there is one more a type of Rodak plate, specific Rodak plate. What is that? It is E type. Now, E type means what? Which organisms we are going to detect in this E type? Enterobacteriaceae. Now, what is meant by Enterobacteriaceae family? It is a family group of organisms which are the organisms which are found in the enteric region of the humans. Enteric region means which region? Enteric region? Intestinal area, gut area, intestinal area. Now, intestinal area means what? There are two types of organism which can be found in this intestinal area. What are those? One, those are the normal flora of the intestine, and second, which are the infectious organism. Now, for example, there is salmonella, which causes. Can you tell me what salmonella causes? It is a
Typhoid.
Yes, salmonella causes typhoid. So salmonella, it is one of the organism from the Enterobacteriaceae family. Then Klebsiella. These are the causative agent or pathogenic organism from this family. There is one more organism, a common, very what do you call? Very important organism or very favorite organism of microbiologists.
E. coli.
E. coli. Very good. So E. coli, it is also the part of intestinal region. It is also from the Enterobacteriaceae family. Now, if there is contamination from these organisms are going to cause some infections to the end users. So, there should be counting of Enterobacteriaceae family members also. It can be Salmonella, Shigella, it can be Klebsiella, E. coli, whatever it is. So, the medium which is used to count the Enterobacteriaceae is called as E type of Rodak media, which is violet red bile glucose agar. So, bile salt is added in this media, which makes it specific for Enterobacteriaceae family members because it is present in the intestinal area. Now, there is next type of Rodak media, which is called as T type. T type means what? Total amount of microorganism. Means it is a general purpose media which will allow the growth of all type of microorganism. It can be any kind of microorganism will grow in this T type of media. It is not a special media. It is not a specific media. So when environmental monitoring is happening in the industry, it can be done by all these different types of media. Few medias I have kept here as an example. So S type of media, which will count the Staphylococcus aureus, which is one of the potential cause of the contamination. Then E type of media, that is Enterobacteriaceae. Again, it is also one of the potential cause of contamination. And next is T type, where all the microorganisms are counted. Now, how much air will be drawn or how much air will be taken for this counting? So generally, in case of active sampling, so active sampling means you are going to force some amount of air on the media. So the amount of air which will be used for this counting is 1000 liters of the air. So with the help of force, the air will be collected, air will be bombarded on the plates, and then plates will be incubated and the colony forming units will be counted. Now, what is meant by colony forming units? So we are going to consider one colony forming unit which is formed after 24 or 48 hours of incubation equals to one cell in the sample. All right, one microorganism in the sample. So if there is one microorganism in the sample, air sample or whatever sample, when that one microorganism settles on the media, and when you incubate that plate for 24 hours, it will grow, it will multiply, and it will form one colony. So that colony can be counted. So it is called as colony forming unit, which will be representation of number of microorganisms present in the original sample. So now every particular country will have different standards for the different industries. So how many colony forming units are acceptable in the air So here I have given one example of Netherlands. So in Netherlands, what is the acceptable limit? So type T, total plate count, means total microorganism in the air. It is how much? Can you tell me? How much is this?
Less than or equal to 2 CFU per.
Less than or equal to 2 means when you are going to count the cells, the colony forming unit should be less than 2 or equal to 2. All right. Now, if it is E, that is when you are counting Enterobacteriaceae, should there be any Enterobacteriaceae family member on the plate when you are sampling there? No, it should not be there. Should there be any S, that is Staphylococcus aureus, present in the plate? No, there should not be any step, because these are the pathogenic organisms. So pathogenic organisms should not be there in the plates. So total organisms can be 2 or less than 2, which is acceptable. All right. So now when you are doing this counting, quarterly counting, how many areas you are going to count this air? So air will be counted from 15 different points, minimum of 15 different points in an industry. All right. So this is how air quality will be checked in the industry. So different areas will be checked, and the air will be either by passive air sampling. Passive air sampling means whatever that Rodak media plates are there, those will be exposed in the area for 4 hours time. Then it will be closed and kept in incubator for the incubation, so that the organisms will grow, multiply, and form the colony. In active air sampling, the air will be forced on the plate, and then the plates will be closed and kept for incubation, and the colony forming units will be counted, and the whatever results are there, those will be monitored. So this is about air quality. Now, how we are going to monitor the surface of the manufacturing area or the laminar airflow? So there are 2 ways to count or monitor the surface. One is by contact plate. So same Rodak plates will be used, and those plates will be kept in contact with the surface of the whatever area you are under monitoring. So the plates will be kept in contact with, it will be direct in contact with the surfaces which you are going to monitor. That is the one way where you are going to monitor the surfaces. What is another way to monitor the surface? By swab sampling. Now, what is mean by swab? I think now during this COVID time, everybody would have understood what is mean by swab, right? So what is swab? Can you tell me? What are swabs? Anybody? What is mean by swab? Have you seen earbuds which you regularly use, right? So you can consider that is one of the swabs. So sterile swabs, that is the kind of earbud kind of equipment we can say, or the particular thing which is sterilized, which will be rubbed on the surface, and then what will happen when the swab is rubbed on the surface? The particles which will be attached, the particles will get attached to the swab, and then that swab will be rubbed on the media. It will be plated on the media, and the plates will be kept for the incubation, and then later on it will be counted for the colony forming units. So surface monitoring will happen by 2 ways. One is by direct contact. The plates will be kept in direct contact with the surfaces, or by swab sampling. These are the 2 ways with which we are going to monitor the surface. Now, what are the next things which we are going to use? Drains and the personal. So drains means how we are going to check for the drains? With the help of swabs only. Now in this picture, you can see this is called a swab. So it is a cotton swab which is sterile, and it will be rubbed on the surface, or it will be used for drains also. and then it will be used on the plates, and then counting will be done. Now, when it is drains, how many areas, how many points we are going to collect the sample? So drains will be considered as critical drains or critical points and non-critical points. Critical points which are very close to manufacturing units, which can cause the contamination source for the product. So those are called as critical points or critical drains. Non-critical drains which are slightly away from the manufacturing unit and which are not in direct contact with the manufacturing processes. So when drains are used for the monitoring process, there should be 29 drain points which has to be monitored. 10 from the non-critical areas, non-critical drains, and 19 from the critical drains. So during the in the manufacturing region, manufacturing area, the 29 drain points has to be monitored on a regular basis. 10 from the non-critical areas, non-critical points, and 19 from the critical points. Now, personal monitoring. How we are going to monitor the person who is handling the equipments or the persons who are present in the manufacturing area. So, personal monitoring will happen once in 6 months. So, once in 6 months, that is half yearly monitoring, it will happen. Now, the sample collection again can happen by 2 ways. What are those 2 ways? One is by the contact plates. So how contact plates we are going to do? The finger dab testing. So on a plate, the person is going to place his or her palm. So all the fingers will be placed on the media and it will be removed, and the plates will be kept for monitoring, incubation, and the counting. Second is with the help of swabs. So when we are collecting the swabs will be collected from the mouth, near the mouth area, armpit area, tips of the fingers, and the palms with gloves, because the person will be always wearing gloves during the processing. Whatever manufacturing is happening, the person will be wearing the gloves. So we have to take the sample swab from the fingers and palm area with gloves, and then the upper surface of the footwear. So these are the points from where the swab samples will be collected from the person. It will be near mouth area, armpit area, finger in the palm, and the upper surface of the footwear. So these are the 4 specimens which are collected, and those are used for the monitoring. It can be air, it can be surface, it can be drains, and the personal. Now, this HVAC validation. Now, what is mean by HVAC validation? I think I have told you, it is heating, ventilation, air conditioning. All these HVAC validation play very important role in the, or it plays a very important role for the end products or the raw material which is used, and the safety of the person and the end user. Now, this HVAC validation will be done in the industry by the external party, external agency every year. Now, what are the parameters which will be used for this validation? First parameter is temperature, relative humidity, and differential pressure. So because in an industry, there are different areas. Different areas is there. Manufacturing area, there is laminar airflow area, clean rooms, pass boxes. So the temperature, relative humidity, and the differential pressures will be counted in these different areas. Then the velocity of the air in different areas, air changes, how there is change in the air in different manufacturing areas. Then filter integrity. Filter integrity means what? The HEPA filters. Can anybody elaborate HEPA filters? What is mean by HEPA filters?
High efficiency
Efficiency
particle
particulate
airflow.
air.
High efficiency particulate air filters. So in laminar airflows or the areas where sterile air has to be inserted or used, all these areas will be equipped with this HEPA filters. So the HEPA filters are used for filtration of the air, to use the sterile air, or to in the laminar airflow area also, to maintain the sterile air condition, HEPA filters are used. So to check the integrity of those filters, once in a year, it has to be validated, it has to be checked. Then non-viable airborne particles and the viable airborne particles. So these are all parameters which has to be checked during the HVAC validation in the industrial processes. All right. Are you getting till now? Is there any
Yes,
doubt?
ma'am.
Is there any doubt?
No, ma'am.
No? So shall we go ahead with the HVAC validation system? Yes?
Yes, ma'am.
Okay. Now, when the validation happens, there are different criteria. Now, there are few criteria which we are just taking a brief overview of those criteria. So those criteria include temperature. Now, temperature of the area, different areas of the manufacturing unit, relative humidity and differential pressure. So temperature will be calibrated, will be counted by the thermometers. Hygrometers are used for relative humidity. So with the help of these instruments, every 2 hours, the readings will be taken. Means what is the change happening in the temperature, relative humidity in a day's time. So 5 readings has to be taken in a day. So whenever they do this validation, HVAC validation for the temperature and relative humidity, so temperature and relative humidity will be checked every 2 hours for 3 consecutive days. Now, what are the criteria for acceptance? How much variation will be accepted? So temperature should not exceed more than 25 degrees centigrade. So what is the acceptable limit of the temperature in the industrial manufacturing process area? It should be 23 degrees centigrade plus or minus 2. So it should not increase beyond 25, or it should not go below 20-21 degrees centigrade. Now, how much is the relative humidity acceptable limit for the relative humidity? Relative humidity can be 45 plus or minus 5%. So relative humidity should be 40 to 50%. Now, if it is, there are areas where the relative humidity will be very, very low. So at lower relative humidity, it can be 15 to 25%. So differential pressure. So DP. What is the DP? It is a difference between the pressure in different areas like production area, surrounding area, laminar airflow area, clean room area. So differential pressure will be counted in the unit of pascals. So the acceptable differential pressure can be 10 pascals across the room. So this is the standard which is given, set by the regulatory body. Now, air velocity. What is the speed of air in different areas of the manufacturing unit? Because the speed of the air, which is going to affect the viable and non-viable particles in the area, in the manufacturing unit. So this air velocity will be calculated, counted by anemometer. Now, how is this anemometer? You can see here, I have put one small diagrammatic representation of this anemometer, which will have the fan. So the fan will be rotating and it will be collecting the air, and with the speed of the air, the fan will be rotated with the speed of the air. So with this rotations of this fan, or those wings of the fan, the velocity of the air will be counted or calculated. Now, this relative humidity and the velocity of the air will affect air changes. Now, what is mean by air changes? what are the changes in the air in different areas. How to calculate this air changes? By the formula given here. So it is always the air change rate, ACH, is equal to the average air velocity, whatever air velocity we are going to count from the anemometer. So average air velocity into the area of the filter, whatever filter is used. So area of the filter divided by volume of the room. From which room you are calculating this air change. So what is the volume of that room? So these are all points which are included to calculate the air change rate or air change velocity. Now this ACH, air change rate, will be calculated for 1 hour, hourly basis. That is the reason in the formula we have multiplied this with 60. 60 means for per hour basis. Now, just an example, I will give you one example. When you put all the things, air velocity, area of the filter, volume of the room into this particular formula, you will calculate the ACH, that is air change rate. If that air change rate is 6, air change rate is coming 6. What is mean by this ACH 6? What is air change rate 6 means? It means the air within that space is completely getting replaced 6 times per hour. Means that is the change in the air which is happening in that area. So air change rate is calculated with the formula given. And if it is 6, means the air in that area is replaced completely every 6 times in an hour. So when air is changing or air is getting replaced 6 times in an hour, you need to monitor different requirements for the industrial standards. So in the manufacturing area, if air is changing frequently, so the monitoring also should happen on that particular basis. So air is changing 6 times in an hour. So every hourly basis when you are doing the monitoring, so monitoring has to be done depending upon the air change rate. All right. So higher values of air change rate, what it indicates? It indicates there is more ventilation. So air is changing frequently. So the monitoring has to happen accordingly. Now, what is mean by filter integrity? Now, HEPA filters are installed in every places for the sterile air. Now, integrity of the HEPA filters, because HEPA filters will, the microorganisms or the particles which are more than 0.3 micron will be bombarded on the filters. So what can happen eventually? It can clog these filters. So the integrity of the filters has to be checked. Now, how is this integrity of the filters, or how should be the efficiency of this HEPA filters? So for class 100,000, the HEPA filters should be 99.97% efficient. Means it should not allow the particles which are more than 0.3 micron, 99.97%. But when it comes for class 100, so ISO, there are different ISO standards given. So according to ISO standards, for class 100 ISO standard, HEPA filter efficiency should be 99.99%. Means it should not allow any particle which is more than or equal to 0.3 micron size in the other area. So all the particles should be restricted on those filters. So this is how is the efficiency of HEPA filters, which will be checked during the HVAC validation. And that's how it has to, if it is not there, if it is not efficient enough, it has to be changed. It has to be replaced. All right. So this is about the general criteria, parameters for HVAC validation. Now we'll move on to non-viable and viable particles, how it will be monitored. So you want a break of 15 minutes. I think in your schedule there is a break. 15 minutes break. Do you need the break or should I continue? Shall we take a break of 15
Yes,
minutes?
ma'am. Yes, ma'am.
Okay. So this next aspect, that is viable and non-viable counting, will start at 11:15, 11:30. Now it is 11:15.
Yes,
Shall we
ma'am.
take a
Yes,
15
ma'am.
minutes break? Okay. Now, just in this break time, just go through what has happened. If any doubt is there, you can ask those doubts when we go for the next session. All right?
Yes.
Okay. Okay, guys. We'll take a 15 minutes break. Is everybody back? Shall we start? Is everybody there?
Yes, ma'am.
Okay. Shall we start?
Yes, ma'am.
Okay. Okay. So till now we have seen what are the non-viable and viable particles. Then the importance of these particles in counting. What are the areas from where the particles will be counted, and the regulations or the regulatory bodies which has given the standards for this non-viable and viable particle counting. Now we are talking about HVAC validation, that is heating, ventilation, and heating, ventilation, all those points where we will be calculating the, just a minute, I think I'm going back. Okay, so HVAC validation, it has to be done on a yearly basis by the external party, external agency, and the parameters for calculation, validation are temperature, air velocity, air changes, filter integrity, and viable, non-viable particles. So we have seen temperature, what is the acceptable range of the temperature and relative humidity, as well as the differential pressure. Then the air changes in the different areas. What are the air change indicators or air change rate? Depending upon that, the specific requirements, adjustments has to be made. Then the filter integrity. So it is for class 100,000, the filter integrity, HEPA filter integrity should be 99.97%. And for class 100, the HEPA filter integrity should be 99.99%. So it should restrict the particles, microorganisms, which are more than 0.3 microns with what efficiency? 99.99%. So that the sterile air condition will be maintained in the area. Now let us come to non-viable particle monitoring. Now, what is meant by non-viable particles? As we discussed before also, non-viable particles are the particles which are in the form of aerosols present in the air, which can be dust particle, pollen particle, any or clothing particle, or any kind of particle which is non-living. And this non-viable particles, non-living particles, what is the advantage? These particles will not spread, they will not grow. But still, these particles will cause threat to the final product, or it will affect the industrial processes if it is not monitored. So this non-viable particle counting will be done automatically. So there are different instruments which are placed in different areas. So the principle for non-viable particle counting is based on light scattering. So during light scattering, a laser beam and the photodetectors are placed in an instrument, and it will detect the change in the light. So when particles are passing through, when any dust particle is passing through or it is coming in contact in the path of laser beam, there will be change in the light, and with which the shadow of the particle will be created. And from that shadow, the counter will detect the size and the number of particles. Now, in general purpose, when it is used, light scattering device, when it is used, it cannot differentiate between viable and non-viable particles, because what is the principle behind this? Laser beam, when a particle, whether it is viable particle or non-viable particle, if it is coming in the path of beam of light, it will create a shadow, and from that shadow, the size and the number of the particles will be counted. But now, shadow will be created by living particles also, living microorganisms also, if it is coming in the path of that beam of light. So it cannot differentiate between viable and non-viable sometimes. Now, to overcome this problem, the fluorescence-induced laser beam is used. Now, this fluorescence-induced laser beam can differentiate between viable and non-viable particles. Now, how this fluorescence will detect or differentiate between viable and non-viable particle? So this microbial particles will, or microorganism will contain some fluorescent particle. So when the laser light, when the fluorescent light is, when the laser beam is bombarded on microorganism, it will induce the fluorescence. When fluorescence is induced in the viable cells or the particles, the optical signature which is developed, with that, the instrument can detect the viable and non-viable particles in the area. So basically, the light scattering system are installed, are used for counting this non-viable particles. Now, in an area, how many places, how many sites these non-viable particle counting will be happening? So the non-viable particle counting or the sampling locations is calculated by the area of the clean room or area of the any particular industrial area. So the locations or non-viable particle locations, NL, is calculated by root A, that is area of the room or area of the clean room. Now, say for example, area of, depending upon this, we will come to know from how many sites, how many places this non-viable particle counting has to be done. So according to the standards, in a packaging room, at least from 5 places, 5 points from 5 different areas, this non-viable particle counting has to be done. While in gowning area, secondary gowning area, where the person will be changing the gowns and entering the clean rooms, in those areas, 2 places, minimum 2 places, from 2 different locations, non-viable particle counting has to be done. All right. Now, this is about the area where how many places, the locations, this counting has to be done. But what about the counts? How many particles are allowed in an area? So for that one, the ISO has given one chart. Now, according to ISO classes, ISO 1, ISO 2, and up to ISO 8. Now, what it indicates, this table indicates what is the acceptable number, maximum number of particles in the air. In particular, how much area of the air per cubic meter. Now, if you see ISO 1, according to ISO 1 standards, there should not be more than 10 particles in the per cubic meter area, of which range more than or equal to 0.1 micrometer size. So the particles which are non-viable particles, which are more than 0.1 or equal to 0.1 micrometer, according to ISO 1, there should be only 10 maximum particles are allowed in that area. What about the larger size, 0.2 or equal to 0.2 micrometer? Only 2 particles are allowed maximum in that particular area. If the particles more the size of the particles, more than 0.3 micron particles are there, that is not allowed in that ISO 1 standard. So these particles has to be monitored and controlled in these areas, industrial processes. So accordingly, you can see here the standards are given: ISO 1, ISO 2, ISO 3, or other standards, class 1 to class 10, class 100. So this particular table indicates, or it gives you an idea of how many maximum non-viable particles are allowed in the environment, in the air of particular industrial processes, clean rooms, etc. So according to these standards, the particle count has to be monitored and kept under control for any industrial processes. Wherever the industrial processes are happening, in the clean rooms, the standards has to be monitored. If the particle count is more than the given standard, it has to be controlled. So there should be controlling devices or controlling systems to control the non-viable particle count in the specified area. So this chart will indicate, this table will indicate how many particles and what size of particles are allowed, maximum particles are allowed in the area according to the different ISO standards. So this is about the non-viable particle count in the air. Now, what about the viable count or viable particles? So you all know that viable particles or viable count includes microorganisms, the organisms which are having potential to grow, to multiply, and to spread. So how we are counting this viable particles? Viable particles will be counted by taking the sample, putting on a media, and allowing it to grow. So you can see here, I have pasted here one photograph of a media. So this is a nutrient media on which you can see this. These are called as colony forming units. All right. Now, if the sample is taken, if sample is from air, it will be directly forced on the plate and it will be incubated, and we are going to count the number of colony forming units. And depending upon that, we are going to check for the standards. But if sample is collected from water, whatever water is used in the industrial processes, if we are collecting the sample or we are checking the viable count in that water, it has to undergo one process, pre-process, which is called as dilution. I hope you would have heard something called dilution, serial dilution. Now,
Yes,
why
ma'am.
we do this serial dilution? Because we don't have idea about the number of microorganisms present in that water sample. If there are many microorganisms in that water sample, you will see crowded growth. When crowded growth is there, you cannot identify this individual colony forming units, and hence you cannot count them individually, which will lead to false positive results. So to reduce the count of microorganism, to reduce the microbial load, which is used for ease of calculation, we need to do something called dilution, which is called a serial dilution. So the original sample will be taken and it will be diluted serially. So 1 ml of the sample will be taken from the source, and then it will be added to 9 ml of the sterile distilled water or sterile buffer or sterile saline. Then that is considered as 10⁻⁻1 dilution, first dilution. Then from that, 1 ml is transferred to the second tube, to the third tube, and to the fourth tube, and so on. So what we are doing, we are reducing this number of the microorganisms serially, right? Now, from each dilution, we are going to plate the sample. So sample will be plated from each dilution on a media plate, which will be incubated, and the colony forming units per ml will be calculated. Now, how is the colony forming units calculated? Per ml, number of colonies into the dilution factor, because now why dilution factor is important? We have not taken the original sample. What we have done? We have diluted that sample. So when we want to calculate the load of the microorganism in original sample, we need to use the positive dilution factor. So number of colony forming units, whatever have developed on the plate, into the total dilution factor, that is a positive dilution factor, divided by the volume of the sample plated. How much sample you have plated? Did you plate 1 ml of the sample directly, or you have plated 0.1 ml of the sample? So depending upon how much sample you have plated, because we have to calculate the colony forming units per ml of the sample. So this is called a serial dilution. So serial dilution is used to count the proper colony forming units, because if the load of the microorganisms is more, or the load of microorganisms are more, we cannot count the colonies in some cases. And when we cannot count the colonies, if colonies are merged, there will be false positive results. So to reduce this false positive results, sometimes we need to do the serial dilution. Not always, depending upon the sample from where you are collecting. So depending upon that, you have to decide whether you are going to do the serial dilution or whether you are going to directly plate the sample on the plate. So this is about the colony forming units for viable counting. Now, how is the standard set in the industrial processes? Now, if we take example of air sampling or the number of microorganisms present in the air, so we can do two ways for the air sampling. We have seen one is active sampling and another is passive sampling. So now what is mean by this passive sampling? Settle plate. Settle plate means what? We'll be exposing the plate, media plate, in the environment for specific time, and then we are closing the plate and keeping it in the incubator, so that the microorganisms which are settled from the air on the plate will grow and form the colonies. So there are two ways. One is settle plate and another is volumetric air sampling. That is the active air sampling, where we are going to force the particular amount of air on the media. Now, when we do this air sampling, what is the criteria for accepting? So if you are doing settle plate exposure, in which areas? In the dispensing areas or in the sampling booth where the sampling is happening or where the product is dispensed. So what are the limits? So there are two indications. What are those two indications? One is the alert. So just okay. One is alert, means when the count is at particular level, you should be alert. And action level means when count is reaching that particular point, there should be some action taken strictly. So you have to be very, very careful, and the action should be taken if the count is going beyond that particular point. Now, under dispensing area or sampling booth, how much should be the alert acceptance criteria? Not more than one colony forming unit. So you should not get more than one colony forming unit. In which area? When you are doing sampling, in the dispensing area or sampling booth area. What kind of sampling you have done? You have done the settle plate exposure. That is the passive air sampling, where 90 mm of plate is taken and 4 hours the air is settled on the plate, and then you have counted the colonies which are formed after 24 hours of incubation. So how many colonies are allowed? Not more than one colony forming unit. That is alert. When you have to take action, again same thing. If there are more colonies than one colony forming units, then some necessary action should be taken in that region. Now, what are the criteria for other locations? Now, as we have discussed before also, there are some locations which are critical, some locations which are non-critical. So In critical areas, there should not be even one, there should not be more than one colony forming unit on a plate. Now, if it is other location, non-critical areas, so when you should be alert when the colony forming units are 75 or above. So you have to be alert. So it means the monitoring has to happen on a regular basis, and some action has to be taken if the colonies are more than 75. There should not be any colony more than 75. If it is more than that, you have to be alert. Now, when you have to take the action, if the colonies are 100, if the colonies are coming, there are no more than 100 colonies, then it's okay. But if the colonies are more than 100, the action has to be taken. We'll see what action will be taken. So if the viable count is in this range, there is alert, there should be alert situation. And if the colony forming units are exceeding some range, some serious action has to be taken. Now, during active air sampling, if you remember, the air will be forced on the media plate for particular time, particular amount of air, and then the plates will be incubated, and next day colony forming units will be counted. So again, under dispensing area, sampling booth, if there is colony forming units are 3 or more, there is an alert. If colony forming units are 5, some serious action has to be taken. Now, other locations like non-critical locations, so the colony forming units are allowed till 100. 100 colony forming units are okay. If it is more than that, there should be some alert. If the colony forming units are more than 200, some action has to be taken to control, to reduce these viable counts, colony forming units or microbial counts. Now, surface monitoring, when you are monitoring the surfaces of the manufacturing area where the entire manufacturing is happening, or clean room is in the clean rooms where research is happening. So in that area, again, during surface monitoring, under dispensing sampling booth, there should not be more than 10 colony forming units. If it is, there is an alert. If there are more than 25 colony forming units, necessary action has to be taken. So it should not be more than 25 under the critical areas. In non-critical locations, non-critical areas, colony forming units of 25 are accepted, but if it is more than 25, serious action has to be taken. So this is how there are standards for viable counts. Now, what kind of viable counting we are doing? If you remember, we have seen that when we use the plates, there are different types of plates we are using. We are using the plates for growth of Staphylococcus. We are using the plates for Enterobacteriaceae family members, and we are using the plates for general, the total bacterial count or total microbial count. So, if the alert limits are different for pathogenic and non-pathogenic microorganism. Now, which are the pathogenic microorganism? If it is Staphylococcus, Klebsiella, Salmonella, these are the pathogenic microorganisms. So the limits or alerts are different for non-pathogenic counts or non-pathogenic microorganism and pathogenic microorganism. So now, how is this alert limit for non-pathogenic microorganism? If microbial count is exceeding the limit, alert limit, what you have to do? What has to be done? So the environmental monitoring, the person who is doing this particular process should inform the quality assurance cell. So whatever quality assurance department is there, the quality assurance department has to be informed about this alert limit. The production head has to be informed, and the monitoring should be continued. So on a regular basis, frequent basis, monitoring should happen. Now, why we are doing this monitoring? Because the limit was under alert limit. So it was not the exceeding limit, alert limit. So when the count is reaching the alert limit, the quality assurance production head has to be informed. Then we need to continue monitoring for this increase in the number of this count, viable count. Then the frequency of the cleaning has to increase, and we have to continue monitoring the count of the non-viable viable microorganisms. So if now during this period, if the count is not increasing, it is consistence. So it is consistently same. So what will be happening? The production can continue. Production can continue if the levels, the microbial count is consistent in the same range. If it is happening, it is coming. So you can continue the production, but some alert is or some information is sent to the quality assurance, production heads, and the frequency of cleaning is increased. So we are going to monitor for the presence of microorganism, and the cleaning frequency has to be increased so that the number of microorganism or the limit will not increase. Now, if it is still increasing, now we have kept the production on, even if the limit of the colony forming unit is under alert limit. Production is on, but what is increased? The frequency of cleaning is increased. And we are doing the continuous monitoring. Now, when continuous monitoring is happening, the number colony forming units are exceeding. Still, it is increasing, and it exceeds the limit. That is the action limit. What should be done? The production has to be stopped. The complete production has to be stopped. Then the information has to go to the production head and the quality assurance, and then production is completely stopped, and there should be investigation happening. What is the source of this contamination? From what region this microbial load is coming? And all products which are manufactured during that time has to be quarantined. So there should not be release of any products if the limit is reached, action limit. Are you getting, everybody? So there are two levels of this monitoring. One is alert limit, when the monitoring is happening for viable counts. So during alert limit, production can continue, but the cleaning frequencies, the monitoring frequencies has to be increased. But if the number of microbial count, viable count, is still increasing further, and if it exceeds the action limit, the entire production has to be stopped, and the products which are manufactured during that time should be quarantined. Now, this rechecking of bio load of all the products has to happen. And the products which are manufactured during the 2 weeks time, before the 2 weeks of this drawing of the samples, should be evaluated for the presence of this microbial load. This action limit is very important. If the microbial count is increasing beyond the action limit, there are very important steps which has to be followed. So first thing is production should stop. The products which are produced during that time should be quarantined, and then before that point, the 2 weeks before those points, all the products has to be evaluated for the same effect. So what are the effects of this product? What are the effects of this microbial load on the product has to be checked. Now, why we need to check this microbial effect of microbial load? Because microorganisms can produce toxins. So we have seen Staphylococcus. So the Staphylococcus can produce toxin. So there are microorganisms which will produce some metabolites which can be causing problems to the product. So all the products has to be evaluated because of the load of the microorganism. So these are the 3 very important steps which has to be carried out if the viable count is increasing more than action limit, or it is exceeding the action limit. The production should stop, the products should be quarantined, and the previously whatever products are produced, those should be evaluated for the effect of this viable count on the products. Now this is about the non-pathogenic microorganisms. So the non-pathogenic microorganism count is increasing. What we have to do? These are the things. But when it comes to pathogenic microorganism, when it comes to, say, for example, Enterobacteriaceae family members, or when it comes to Staphylococcus or any other pathogenic microorganism, there is nothing like alert limit. So the production should stop immediately. The production head, quality assurance people should be informed about this particular presence of pathogenic microorganism. All products should be quarantined during that time. And again, the same thing, the rechecking of the load, which has happened previously, 2 weeks before the presence of this pathogenic microorganism. So rechecking of those bioloads to evaluate the effect of this microorganism on the product should happen. So this is about the action taken for viable counting. So what action we have to take when there is a presence of pathogenic and non-pathogenic microorganisms present in the area, whatever area it is. So during this particular time, there are different standard operating procedures for clean room, sanitization of that particular area. So all those standard operating procedures should be followed to control this load of microorganism in the industrial manufacturing area or the processes. All right. So this is about the viable and non-viable count, or the fundamentals of viable and non-viable counting in the industrial processes. Now, with doing this all counting or viable and non-viable monitoring, what we are going to achieve, or what is the importance of this monitoring process? So the first thing is control on the quality. So by counting these particles, we are going to check, we are going to maintain the quality of the processes in the industry. So we are going to meet the standards which are specified by different regulatory bodies. So every regulatory body would have given the standards. They would have ensured that the standards should be maintained. So by checking this particle counting, we are going to ensure the quality of the products, and we are going to see that the products are free from any contaminants. So for the quality of the product, this particular aspect is very important. Second thing, regulatory compliances, because industry has to follow the regulatory compliances given by the regulatory bodies. So we have seen FDA, ISO, PDA. These are the regulatory bodies which will be giving the standards for this viable and non-viable particles. So by counting, by monitoring this particle count, we are maintaining the strict regulatory standards. So whether all the standards, all the regulations are maintained, which will be used for safety and efficacy of the products. So we are maintaining the quality of the product. We are maintaining the regulatory compliances. Then process optimization. Now, if there are any viable, non-viable counts, it means the efficiency of that particular process is low. So there will be wastage of the products, because if there are viable and non-viable particles present in the product should not be released in the market. It should be stopped. It should be seized. So what is happening? It is getting wasted. So the process will be optimized by this monitoring system. We are going to optimize the manufacturing process, identifying, mitigating the sources of the contamination. And by doing this, we are improving the efficiency of the product. And obviously, we are reducing the waste which is getting generated because of presence of this viable and non-viable particles. Next thing is in terms of health and safety. So by monitoring this particle count, or this monitoring particle count is very crucial in maintaining the healthy and safe environment for the workers as well as the consumers and the general public, because we are monitoring the drains also. We will be counting the waste which is going out of the industry. So this particular monitoring is going to help, or it is going to play a crucial role in maintaining healthy and safe environment, not only for the workers, for consumers or the end users, and also for the general public. All right. So this is how the environmental monitoring of viable and non-viable particles in an industry help for all these processes, whether it is quality control, whether it is regulatory compliances, whether it is process optimization, or whether it is healthy and safety environment for all the workers, consumers, and general public. So this is about the viable and non-viable particle counting fundamentals and the importance. I think we have come to the end of the session. Now, if you have any doubts, any questions regarding this viable, non-viable particle counting, please you can ask. Go ahead and ask the doubts. Is there any doubts? Yes. Is there any doubt in this viable, non-viable particle counting?
If nobody has any doubt, means we can end today's session. Thank you, ma'am. Thank you for your time. So
Thank you, everybody.
we'll meet tomorrow. Okay.
Thank you, ma'am.
Thank you, ma'am.
Thank you,
Have a
ma'am.
nice day.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, ma'am.